And what to keep in mind in molecular docking is all the things that we are going to see. We are going to see two tools, one which is very popular, which is Autodoc. Then another one, which is a web-based tool, which is HadDoc. So I will show you both and I'll tell you how to do the docking. Okay. So let me share my screen. Okay, so basics I'll start with. Is my screen visible to everyone?
Yes, sir.
Okay, so we're going to see what molecular docking is, introduction, its methods and application. So at this point of time, since many of you come from different backgrounds, so I want you to imagine something that I'm telling about. Okay, so imagine you are in a house. Okay, and then in a house, that entire house is just one big unit. Okay, there's no bedroom, bathroom. It's just four walls. Okay, imagine something like that. And inside that, imagine about 100 or like 50, 60 people standing and moving around. Okay, so there is some space, but for some people, they are crowded in one particular place. There's not much space. So it's a small house, and there is a lot of movement happening among all of them. Now, when you're imagining this scenario, what all things would happen? You will see immediately that there is never a situation when one person is not going to be in contact with someone else. Someone will be like their arm will be hitting someone, they will be coming face to face with each other, or someone will be like, if let us imagine there are kids, then elder people are holding the kids together and taking them around. So there is a lot of movement happening, there is a lot of interactions happening, and there is a lot of collisions happening. Okay. Now some collisions are transient, some collisions are permanent. Like for example, in that same example scenario, imagine a mother and child. So that interaction between the mother and child, wherever the mother is going, the child is also going with the mother. Then you can imagine two friends. So they will come for 5 minutes, talk with each other, and then they will move away in that same house. After some point of time, they'll come again. They will talk about some topic, and then again they will move about. Okay. Now, as I said, it's a crowded room. There is very less space. Everyone is moving at some point of time, and there's always a movement. They are not moving in the same orientation. They are moving in different orientation.
Sorry
In,
for interruption.
yeah.
Kindly explains what is rational drug design.
Yeah, I'll come to that. Yeah, I'll come to that. Good question.
Thanks.
So now instead of this imaginary house or imaginary room, this is a cell. Okay, now replace that imaginary house with a cell. And in the cell, instead of people in the house, you imagine macromolecules or molecules that are moving around. This is how a cellular component in the cytoplasm of either a prokaryote or a eukaryote is happening. So they all are moving around, they all are having some collisions. And even within the cytoplasm, there are places where interactions between two molecules are permanent. Interactions between two molecules are transient, like the case of mother and child. Let's say a protein which is bound to DNA, it will be always bound to DNA, and then after some point of time, it will go away. So, like the child moving away from the mother for playing and then coming back. Similarly, two friends talking about some topic and then for some period of time moving away. So the same way, you can think about biomolecular interactions is what is happening inside the cell. And there's a lot of interactions that happen. And these are important. Now, in the cell, these interactions drive the entire physiology, the cellular process, pathways, and everything inside the cell. At one point of time, when computational power came to be seen as a driving force in biology, and many different data, especially three-dimensional data of proteins and molecules were available, people started asking the question, how do these interactions happen? Or how can we predict certain interactions happening between them? So, in the case of molecular docking, you are using a computational method to identify how two molecules are interacting with each other. Okay. Now, the most common application these days for molecular docking is for drug design. Now, in the history of drug design, we had many situations where molecules or active components were identified serendipitously. For example, for malaria, they knew that the tribal areas, they will go to the quinone tree or the cinchona tree, and then they will take the bark and start chewing it when they get the shivers. This was a traditional folklore or indigenous oral tradition that was there prevalent in the tribal areas and wherever malaria was very endemic. Now, when malaria was identified as a vector-borne disease, the identity of quinone was identified because one of them started testing this particular theory that there is some active component in the bark of the tree, and then they found it that actually is very effective in preventing malarial disease. So, and the same another example we can say is how Alexander Fleming identified penicillin. He was gone on a vacation. He left some plates, agar plates, and then when he came back, he's found zone of clearance in those places wherever there was a fungi that was grown, a mold that was grown. Later on, he isolated it and he found it as penicillin. Now, all these identification of drugs through these mechanisms, where it happened by chance, or they listened to some traditional, already present knowledge, and then they applied it. So, those things we now call it as, within quotes, irrational drug discovery, because they did not know what the molecule is that is isolated in the bark of a tree or from a fungi. They did not know what these molecules do in the pathogenic bacteria or the parasite. They just know that it worked. So it was prescribed as a drug. There were a lot of instances, there are a lot of other molecules that also had similar history. Now, with 1950s and 60s, when X-ray crystallography became more prevalent, structures were identified. Some high-profile structures of like cholera or other viral diseases were identified. Suddenly, now people started talking about molecular interactions. And the molecular interaction is when you have the structure of the protein and you have the structure of the molecules which are supposed to prevent the function of this particular protein or this particular target. So those type of studies where we know both the structures of the active component and the protein, we call it as a rational structure-based drug design. So rational, the word rational here is a bit misleading because in the earlier one, when Alexander Fleming discovered penicillin and how penicillin was extracted and penicillin was found to be an active component in eradicating bacterial diseases, is also rational. But the rational here is that there is a scientific or a logical step. Maybe we can probably call it as more logical because you know the structure of the both the molecules that are playing a role. In the case of the other types of drug design, where you know the host target, but you don't know what phytocompound is working against it, then in that case, you cannot identify what could be the compound that is causing the inhibition. Similarly, you know the extract works, you know what are the components of the extract, but inside the human body, we do not know which particular host target those compounds bind to. Again, if you draw a grid where on one axis you say protein structures, yes, no, and on another axis you say ligand structure or molecule structure, yes, no. In rational drug design or structure-based drug design, you need to know both the protein structure and the small molecule or the ligand structure. You cannot go ahead with only one structure in the rational drug design. That being said, there are other types of methods like receptor-based pharmacophore or ligand-based pharmacophore or QSAR studies. In all these studies, you do not have one particular type of structure. Either the target structure is not there or the small molecule structure is not there. So in rational drug design, you need to know both the structures. Now, when you know both the structures, the next question comes is how do they bind? Okay. We know why they bind because they are involved in some kind of activity, but how do they bind? And if let us say you have 1 million such compounds and you want to find the best binder among all of these, then you need to have an automated method of looking at all the compounds and finding out which is the best. And that is the virtual screening, which is part of the molecular docking. So, what is docking? Here we are predicting the optimal physical configuration and energy between two molecules. So that is the main idea. So I have, for example, the figure of Shrek and donkey. Now we know from these two figures that Shrek is a bit larger than the donkey. Now the canonical view is that Shrek used to carry the donkey with him. Okay. And in this particular case, the donkey is, you can say, docked with Shrek. Okay. But that is not the only one. But you can actually have a way in which donkey is like this on Shrek, okay, on his shoulder. That's also docking. Okay. Now, in this, I have shown two confirmations. One where donkey is being carried by the Shrek in his arms or on the shoulder. Now, which one is optimal? Okay, so if I go by the strict canonical knowledge, based on the literature and the visual movie, I know that Shrek used to carry donkey like this. So this is probably optimal in this particular scenario. So, but then in the case of biomolecules, how do you know which is optimal? Okay, you know that molecules can bend, can rotate, proteins are not static, they are very dynamic. Then how do you know which one is optimal? So molecular docking for a non-biologist is nothing but an optimization problem. Optimization problem is a branch in computer science where you have multiple solutions to the same problem. But not all solutions are optimal. Only one solution or maybe two solutions are optimal in that. So in this optimization problem, you can employ different types of algorithms to identify this optimal solution. Those who have some bioinformatics background have done some sequence analysis. Dynamic programming is also an optimization problem because you have many different solutions for two sequences that can align with each other. But the optimal solution in the alignment is the one that has the maximum score. So there is only one solution or maybe two solutions available. So the docking problem, it orients the two molecule in a way thereby the interaction is maximal between them. So take the same example of Shrek and Donkey. In this particular case, I know the interaction is maximal. The entire body of Donkey is carried by Shrek. But in this particular case, probably not so, like where he is carrying on his shoulder. Okay. Or Shrek could be riding the donkey. Again, that is not a very optimal interaction between them. Now, among all this, when you have a maximal interaction, there could be a scenario where it may not have the lowest energy. So you want to find the lowest energy or the total interaction energy such that the binding energy is minimum. Now, going back to the same example that I told yesterday, you have among an acrobat or a contortionist, you have many different ways in which the human body can move and rotate, and there can be a lot of different ways they can twist and turn. But there are two conformations in which we have the lowest calories burnt, which is one is sitting and the other one is sleeping. So, in the same analogy, even in molecules, if two molecules are optimal and making interaction with each other, they will have the lowest binding energy, meaning that their energy, total energy will be very low. If it is high, that means they are not happy with each other, they will move away. And docking tries to identify what structural changes are brought about by the interaction. Okay. So, as I said, you have two objects, two entities through three-dimensional objects. You are bringing them together to find out what is the interaction between them. So, in the drug design terminology, we say a ligand and a receptor. Now, in many places, you will hear the word ligand, small molecule, drug, phytocompound, or active component. All these refer to the small, the drug-like property that the molecule has. And the other one is receptor. And you will hear words like receptor, protein, target, drug target, or host target. All those refer to the molecule that to which this ligand is binding to. So for most cases, I may be interchangeably using these terms, but understand that you have a ligand and you have a receptor, and this ligand receptor together they form a ligand receptor complex. We want to find this complex which is optimal. Now, one thing that will pop up to you in this screen, and even in the example of Shrek and Donkey, is that Shrek is a bit larger than Donkey. Similarly, the green object here is larger than the red object. Receptor is larger than the ligand. Now, so automatically, what we are doing is not we are not docking the receptor to the ligand, but we are docking the ligand to the receptor. So the relatively smaller sized molecule is docked to the relatively larger sized molecule. So when you take a larger sized molecule, like let's say a protein, here it is a rapid muscle GPP, you will have many different places in which the molecule can bind. It can bind to the glycogen storage site, it can bind to the inhibitor site, it can bind to the catalytic site, it can bind to a allosteric site, or some other place. So which particular place does it bind to? Now this is a question by itself. Now this question is nothing but where does it bind, right? So we instead of how it binds, we can rephrase it as where it binds. Unfortunately, molecular docking is not used to answer the question where in the molecule, the small molecule or the ligand, or where in the receptor the ligand is most preferred to bind. We don't try to answer that question. You know where it binds to a large extent. You have a lot of evidence for it, and then you're trying to bind how it binds. We don't even ask the question why it binds because there is some evidence to it. We just want to answer the question how. How does a small molecule or a ligand bind to a receptor? That's all. Where it binds is something that needs a lot more evidence, and you need to do something called as the active site prediction in the receptor to identify which particular site in the protein or the receptor is more amenable for something to bind. As a general rule of thumb, proteins, when they have a shallow or a cavity region where the solvent is accessible and when something binds and the solvent can be removed, is occluded, those regions are amenable for something to bind. Okay. So active site prediction problem, that's a separate topic by itself, tries to solve where the protein can have regions in which small molecules can go and bind to. So what is the difficulty in molecular docking? Seems very straightforward, right? But the first difficulty is molecules are very flexible. Take the same Shrek example. Shrek can move his arms, he can move his legs. Shrek may not have his arms in a position that he can carry the donkey. His arms have to be extended out to carry the donkey. But so molecules are highly flexible, proteins are highly flexible. They don't stay in the same orientation for a long periods of time. They keep shuffling between intermediate states and so on. So this goes against all the textbook type of knowledge and high school and lower undergraduate knowledge where we talk about is proteins are present in only one particular conformation. That's not true. So even if you take an X-ray crystallography structure, even though we see only one structure in the X-ray file, in the PDB file, you can generate thousands of conformations because that one structure is an average of all the molecules that was there in that particular crystal. So it's an average model. So average model means that there are multiple conformations, and if there are multiple conformations, then it is very difficult to find out the electron density. So molecular flexibility for the protein itself is a big limiting factor. And the same thing happens for small molecules or ligand as well. Ligands are highly rotatable. Okay. So when the same rotational bonds that we saw yesterday, which Ramchandran found out, the psi-phi angles, the more number of rotational bonds are there, the more flexible the molecule is. So a highly flexible molecule may not have one single conformation at the lowest energy state. It can have the same energy level and it can be in multiple different conformations for a ligand. The second difficulty is thousands of degrees of freedom. So every rotational bond adds a degrees of freedom. Now, what do we call as a degrees of freedom? I don't know if someone has explained this in the earlier lectures. So let us imagine this particular case is a three-dimensional object. Of course, it is. Now, what are the degrees of freedom for this? Because this entire case is a one single entity, I'm going to assume it's having a it's like one solid shape. It has 6 degrees of freedom. It is a three-dimensional object, so it is in XYZ coordinates, and I can move on the x-axis. If you look at my video, you will see I'm moving the hand. It's on the x-axis, I can move. This is called translation. I can move on the y-axis and I can move on the z-axis. So there are 3 degrees of freedom for translation. Okay. Similarly, I can rotate it in the x-axis, I can rotate it in the y-axis, or I can rotate it in the z-axis. So I have 3 rotational degrees of freedom. So for one three-dimensional object where it is considered completely rigid, there is no rotational bonds inside. Okay, you have 6 degrees of freedom. Now let's add a complexity to this. I opened the lid of this airport, and now this has a rotational angle. Okay, so this I can move it in many different ways, and I can have it fully open or half closed or completely closed. I can have multiple intermediates of this, and in each of them you have 3 degrees in translation, 3 degrees in rotation. So when you have multiple rotational bonds or multiple angles, uh, torsional angles as we call it, you add more complexity, you have more flexibility. So for any small molecule, you will have at least 4 or 5, at the maximum 30 rotational bonds. So that adds thousands of degrees of freedom. So when we are taking both the flexibility of the receptor and the ligand, then you have number of combinations is astronomical. So how do we do this? How do we solve this problem of finding the optimal configuration? So the first way we do it is we do rigid docking. So we keep the donkey rigid, we keep the Shrek rigid, and then in a three-dimensional space, I'm rotating the donkey, I'm translating it as I rotate, and I'm placing it on Shrek's hand. That's rigid body docking. Shrek. There I go. Okay. Right. So what did I do? I did not move Shrek. I just moved donkey. Okay. So this is very easy to do. And this solves a lot of things. So you only have 6 degrees of freedom, only for one molecule. And you do not take into consideration flexibility of the molecule that you're moving. And you can examine energetics of the possible binding conformations. So you have protein-protein docking with rigid body movement. You can have protein ligand docking also in rigid body docking. So the next type of docking in protein ligand is where you make the the receptor rigid. Shrek is standing here. But as I'm moving donkey can also move his legs, and there is some rotational degrees. You can see his legs can move. And so you have different types of conformation in which donkey can be placed with the Shrek. So you get multiple solutions. So in this particular case, the number of search space, that's what we call it, the search space with which I'm making different conformations of donkey with the 4 rotational bonds he has for each of its leg. You have different types of conformation. So that's a search space. So imagine the search space is also like a valley in a geographical setup where you have hills and valleys. So one confirmation may be at this part of the hill, another confirmation I move a leg, he will be probably here, another one on this side of the hill, another one on this side of the hill, and then somewhere here. So you have a large confirmation search space. So you can use some Monte Carlo or some kind of methods to identify the search or the conformational search. You also have, depending on how many molecules are there, how many time that's taken, you can have rough docking or detailed docking. Rough docking is where you take the molecules, you quickly see if the molecules can uh, uh, have a conformational change and they can bind or they don't bind. So it's a very quick way to eliminate bad binders. Whereas detailed docking is where you are looking at the specific interaction, you are looking at uh, how these search space can be explored in detail, and this can be done only for a couple of molecules. You can't do this for 1 million compounds that you want to screen. So there's a question in the chat box, how to determine the rigidity and flexibility of protein. So there are tools where you can do the conformational search, or you can do a conformational ensemble of the protein for any given protein. The easiest way that you can do is you go to PyMol. Let me see if I have a structure. I can show. The very easiest way you can see. So this is a protein structure. Okay, this is shown in cartoon representation. How do we know which particular regions of the protein are highly rigid or highly mobile? The easiest way is to look at the B factor column. So in the protein structure, uh, Yeah, look overview and so if you scroll down in your PDB file, I hope does everyone know what PDB file is? What is a PDB file format? Yes, no.
Yes, sir.
Okay, so then in that case, you would know that this particular column here, the number 11.88, that particular column is the B factor column. The B factor ranges from a small number to a high number. The more higher the number, that means they are highly flexible. That's the temperature factor. So in PyMol, you can visualize those temperature factors by show no preset B factor putty. So here immediately you see that these regions are not moving a lot. The B factor is low. Here, the B factor is very high, especially for this particular one or two residues here is showing in red. So this is the region where it is highly flexible, whereas these regions are not so flexible. So this is one way of finding out rigidity and flexibility in proteins. The other way is that you put this protein in a solute box in water, you give a force, and then you see how it accelerates. So basically, you're solving the second law of Newton, F equals ma. You know the m because we have the atoms, so we know the mass. We are giving a force. That force is through temperature. So, like for example, 330 Kelvin. And at the temperature, what happens to the protein? So it will show some conformation. So certain regions will move a lot, certain regions will not move a lot. So you can find out which regions are rigid and which regions are flexible. So that particular technique is called molecular dynamic simulation. But then there are other tools also available where given a protein structure, it will identify rigid parts and flexible parts, and it will tell you which is the hinge region, which makes it flexible and uh, rigid. Did that answer your question?
Yes, sir. Thank you so much.
Okay. So, as I said, you can screen good binders or eliminate bad binders in rough docking. And you can look at how each binder is working in a particular way by doing a detailed thermodynamic calculation and all that. Okay. So you cannot do this for a million compounds. You can do this for a million compounds. Okay. So this is the entire drug discovery pipeline from target identification validation to uh, uh, pharmacology synthesis and clinical trials. So in between, you have virtual screening and structure-based design, which is where you have molecular docking coming into play. And this is the reason why molecular docking is used in rational structure-based drug design. The rationality, as I said, is you need to have both the structures available, and that creates a logic. You have a logic as to how they bind to each other. So what do you need to know for docking? The structure of the receptor, whether the structure is relevant for what you're looking for, what ligands you're looking that you want to bind to the receptor, and the mechanism of the binding site or where you want to bind it. All this you need to know. Now, in many cases, this particular last bullet point may not be available. You may not know where it is going to bind. In those particular cases, you do something called as blind docking, where you give the entire protein itself as an active site. You allow the small molecule or the ligand to bind wherever it wants, and then you see which is statistically significant in binding to a particular area, and then you see whether that is having a function or not. Okay, so most of the time we do site specific docking. We know where it is supposed to bind, what type of mechanism it is doing, and that is where we try to do the docking. There are different methods and different algorithms used. As I said, we will see a little bit about Autodoc. Autodoc uses Lamarckian genetic algorithm and simulated annealing to find the conformational search space. So the conformational search space is not for the receptor, but for the ligand, because the ligand has highly flexible. There is a lot of conformations that it can take. So you are trying to see which conformation is best suited for binding to the receptor. Um, so this entire field of tools for developing molecular docking and virtual screening started somewhere in the 80s. Doc in University of California, San Francisco, Irwin Kunz, his group is still active. They came up with the program Doc, where they did rigid body docking. That was all the capacities or the computational resource that was available. But as the years went, memory chips became cheaper, storage became cheaper, and computational power increased. And we now have a whole gamut of tools that actually do a lot. And we have proprietary software from Schrodinger or Gold or FlexX, which can do a lot more new types of docking, right? Or uses much better approaches. List of tools. Some of these are good, some of these are very specific to what you want to do, and some of them are proprietary, like Glide, Gold are proprietary. They are not freely available. They cost a lot of money. So one of the easiest ways you can think about how docking works is that you have, let us say, this molecule and this bond here, which is connecting this five-membered ring with the carboxyl group and the amine group. This particular bond is the rotatable bond. So basically, this is the bonds within the five-membered ring are rigid; they can't move. The bond between the amine and bond, the double bond O, can't move much. This is the only rotatable bond. Okay. So now what you can think of is that you can have a conformational search space where you have 360 different conformations, because you can have this rotatable bond turned 1 degree to each. And so you have this particular structure getting a lot more conformations. In the receptor, this is the gray area is the receptor, the protein that you want to bind to. You see that where there is a particular region which is amenable for either this part or this part to bind. So if it is a hydrophobic patch, that is a hydrophobic patch, it is makes a lot of sense to have the ring structure placed there, because most of the time there will be cation pi interactions or pi stacking or some kind of hydrophobic interactions happening between them. And since there is a oxygen here and a hydrogen, these are hydrogen bond donors and acceptors. And if you have a charged area in your protein surface, then you can place that. Now you have these two parts of the ligand, which is not connected by this bond, now placed in particular regions in the protein where they are most likely go to bind or going to present. And then you can build the bond between them. Okay. This structure and this structure are not the same, but I'm giving you an example as to how fragment based docking is done. This is called incremental construction. So you fragment the ligand into rigid and flexible parts. You place the rigid parts first, and then you try to build the connection between them. So incrementally, you try to do one at a time. So this incremental construction is what doc does. What doc does, and it's very successful in finding for many different small molecules. Where it fails is that if your molecule has too many rotational bonds, more than 30, then you will not be able to find the region where it will bind. You will be having multiple answers, and each of them is possible, and there are multiple solutions to it. Um, so you can have different shape matching algorithms. You can have complementary ligand-based matching algorithms. So there are different algorithms. All these algorithms are trying to do is doing the conformational search space. Okay. So docking, the take home from this lecture, if it is that docking involves two steps. The first step is finding out the conformational search or the space of the flexibility of the ligand. And then the second one is the scoring of how good that conformation is that's going to bind. So the conformational search is what most of these genetic algorithm, um, incremental search, taboo search, or some kind of shape matching, all these are doing is finding out the complementary shape or the conformational search space. You also need to understand in the scoring, the second step in molecular docking, once you have found a conformation, you want to bring it to the receptor and then find whether it is a good binder or not. That scoring involves entropy and enthalpy terms. It's a thermodynamic process. And in the thermodynamic process, you have to understand that the receptor would not be waiting for the ligand to come and bind, but the receptor would already have been present to bind with some water molecules, and the ligand displaces these water molecules and makes the same interaction with the receptor. So the displaced water, the energy required to bind these water molecules is what is calculated in the scoring function. Just like how you have different conformational search algorithms, generic algorithm, taboo search, um, incremental construction, shape matching, and so on. You have different types of scoring functions also. All these scoring functions, the way they have come up with is through empirical data, meaning through experimental data. There would be some free energy calculations that could have been done. There will be some uh, binding displacement terms. There will be KD, KI terms. Using all these terms, they will come up with a scoring function. Each scoring function has its own advantage, disadvantage. Some scoring functions are very good only for small molecules. Now, small molecules is a term that's used in drug design, where if the molecular weight of the molecule is uh, about 500 daltons or less, that's called a small molecule. Anything more than that is not a small molecule. In some cases, those scoring functions would be very good for DNA also. In some cases, it may be good for peptides as well. In some cases, the scoring functions may not be apt for peptides or DNA. So it depends on the scoring function, depends on what is being used. So depending on the scoring function and depending on the conformational search, certain algorithms do a good job of finding conformations between two different molecules. Some don't do it, some do it only for certain molecules. Okay. Let's see if because we're already 3:48, I'm going to okay. So let me talk about this particular part. Yes, please go ahead.
Sir, I just want to check my understanding. So, in proteins, there are some rigid part that act as a catalytic site. So,
Yes.
catalytic sites may be single or multiple.
No, catalytic site is a spatial region. Okay. Okay, spatial region meaning that it's a three-dimensional space. And in the three-dimensional space, you have a particular area in the protein where something can go on by. That is the catalytic space or the catalytic site. The catalytic site is made up of multiple residues, multiple amino acids. So in the case of serine proteases or some other type of enzyme, they have a catalytic triad, meaning there are three residues that come closer. They are not sequentially in close by in the sequence, But in a three-dimensional space, they come closer and they are present as a triad. And it is in that particular place that it does its job of cleaving the bond. So that particular catalytic site is made up of three residues. So that active site or catalytic site is the made up of three residues. Now, the question, the thought that this particular site is rigid comes from the theory or the hypothesis that you have the lock and key mechanism. You have different types of locks, you have different types of keys, but you have only one type of key for every lock. Okay, so the space that the lock or the key occupies inside the lock is predetermined. So an exact match can be found and you can find a key to that particular lock. Okay, that is the lock and key mechanism. But lock and key mechanism is not valid. It assumes a lot about the protein structure. The mechanism that we now know because of protein conformational flexibility is that proteins can go in an open and closed conformation. So that region that it wants to bind or it binds to something may be not in a open form. It may be closed, and when the substrate comes closer to it or the molecule comes closer to it, it opens up and binds to it. So the induced fit model, in which we see in enzymes, that to a large extent happens with highly flexible proteins. Invariably, you have intrinsically disordered proteins where the active site is not defined until it comes in contact with the substrate or the ligand. So it is at that point the entire disordered part of the protein becomes ordered and you have a conformation of the protein. So there are a lot of theories that have been found outdated, and the lock and key mechanism is pretty easy to understand because it's very simplistic in terms of a protein that has a shape and you have a ligand that complements the shape and it binds to that particular place. So the rigid body docking assumes the lock and key mechanism of the receptor and ligand binding. Did that answer your question?
Yes, sir. So hypothetically, if protein structure have helices or beta sheets and fold, so depending on the space present in between them, only that region can be act as a active site.
Not necessarily. In as a general rule of thumb, as I said, any protein, like for example, if this is a protein and inside this the region which is a cavity, I don't know if you can see it. That's the region where I can put my headphones. That is a cavity, and that is amenable for something to bind. Okay, whether I put the headphone or I fill it up with water, there is a space for something to bind. Okay. Um, in many cases, the active sites are not defined. For enzymes, it is defined. For structural proteins, you don't have an active site. The term active site or catalytic site comes only for proteins where they do have a catalytic mechanism. Or, for example, DNA binding proteins. They bind to DNA. So the region where it binds to DNA, that can be termed as an active site. Okay. But basically, those are regions where it is binding to the substrate or the ligand.
So catalytic enzymes are enzymes are different and structural proteins are different, sir. Both are different.
So let me show you this slide. So proteins can be can do catalysis. Proteins can be structural, actin, myosin, keratin, collagen. It can be used in transport. They can be hormonal proteins. They can be defensive proteins. They can be motor proteins. They can be storage proteins. They can be receptor proteins for signaling. Most of the drug targets are GPCRs. There are a lot of drug targets which are structural in nature, and there are a lot of drug targets which are motor proteins in nature. So, what type of protein you are trying to target may be one of these categories. Understood? Proteins come in different shapes, size, different, there's a huge diversity of proteins. Which particular protein is a target, which particular protein is not a target, is the entire part of target identification and validation, which comes at a very early part of the drug design cycle here. Actually, target identification and validation. Right. So that part itself is a topic by itself. How do you know which particular protein is uh, uh, can be considered as a drug target, which particular protein is not a drug target? How do you decide that? That depends on this particular step. Clear?
We have a separate talk on that, drug target
Yes.
identification.
It's a separate field itself, not just a talk. I mean, I understand in this course, but there is a lot of, there is a whole concept of druggable genome. You take the malarial genome, you take a filarial genome, or any parasite that causes a disease. How much of the genome is druggable? Meaning, how much of those genome you can identify targets, and how many of them you can create drugs against it? That itself is like one full PhD. Okay. So, receptor flexibility, this actually uh, is something that most of the tools are now taking into consideration, because they know that the lock and key model, or if you keep the receptor rigid and only move the flexible ligand and dock it, that is just half of the story. You don't have, uh, you're not taking into consideration the receptor flexibility at all. So in Autodoc, Gold, and even in Glide, you have modules where you can make certain parts of the protein flexible. You can't make the entire protein flexible because that is probably computationally very intensive. And there are other complementary methods that can be used to do the same thing. But for docking, the region where the active site, so for example, the residues that are involved in the active site, if you make that region alone as flexible, then it kind of answers a lot of question, and it takes a lot of things into consideration. So there's a lot of advances that has happened, and with AI tools like diffusion models, you don't even have to consider the ligand flexibility or the receptor flexibility. You are creating, using a diffusion model, the type of molecule that may be present. So Rosetta diffusion, RF diffusion is a pretty good um, tool to do that, right? So having said all this, what I'm going to show you is, which is relevant to your topic, the virtual screen. Now that, let's say you are doing molecular docking and you're screening 1 million compounds or 100 million compounds, that particular process is called virtual screening. So virtual screening involves docking, but you are looking at certain terms, something called hit rate. What is the recovery of true positives in the data set or the molecules that you're taking? How many of them did the docking method identify as like really good molecule, good binders? And then enrichment factor. Let us say you do different types of docking. You do rough docking, then followed by detailed docking. Did the number of active compounds increase in the subsequent steps? Did you remove more bad binders from the second or the third step? So those types of parameters are seen and identified to actually see which can be a good drug molecule. So success and failure of docking is one of the easiest way to identify is through root mean square deviation or RMSD. Let us say you are targeting a receptor that is a enzyme. This enzyme binds to a known substrate. Okay, let's say glucose. It binds to glucose. Right? You are designing a molecule that is similar to glucose, and you are docking it. If and only if you have a crystal structure or an experimentally solved structure of the receptor with the glucose bound in it, you can do this RMSD comparison with the docked compound, and then you can see whether that particular compound conformation is very similar to the glucose conformation that is bound to the receptor. So the RMSD, as low as it is, if it can be less than 1 angstrom, then you can say that the binding mode is closest to the native substrate. It makes sense. Think of this analogy. You have um, a person, let's say, I can think of only a bad analogy, but it's a good analogy for this particular instance. There's an exam that's going to happen, and there's one person who has to write the exam, but then they don't know the subject. So there is another person who looks like them, and then they impersonate that person to write the exam. Okay. So in that particular case, the invigilator may not know whether it's the same person because, like, for example, if they are identical twins, it's absolutely the RMSD or identity between them is so high that you can mistake one twin for another. So if that is a very good. So similarly, if a molecule that has been bound with the receptor binds in the same binding conformation, binds with the similar binding energy to the receptor, like the native substrate, then that is a success because you have found an alternate inhibitor or an alternate molecule that binds in a very similar fashion to the native substrate. So there will be competitive inhibition when you are in the real world where you are giving both the substrate and the small molecule. So ranking of the energies based on the lowest energy score is also a very successful way of finding out. So as I said, you want to find one that has maximum number of interactions. Maximum number of maximum number of interactions and has very similar binding energy and the lowest energy, meaning very low number present kilocalorie per mole compared to the known molecule. For example, the same example, glucose and glucose-like molecule. If glucose binds with -8 kilocalorie per mole, then your molecule should be at least -10 kilocalorie per mole, at least 2 kilocalorie per mole, or much lower than that. Then you will have a competitive way of binding with the known molecule. So in molecular docking, just like how you do wet lab experiment, you need to have controls. The controls can be both positive and negative. Positive control is where you know it is a molecule that binds to the receptor. Same example, glucose. So you can find the binding mode of the glucose, you can find the binding energy of the glucose. So there's a positive control. Negative control is where you know the particular molecule that doesn't bind to the particular receptor. Like for example, xylose doesn't bind to it. Okay. So that particular molecule, if it is very close to xylose, then they will be agonist and not a antagonist. So all these different things are something that need to be taken into consideration when you have finished the docking. So doing the docking is not the big thing. After docking, the analysis, the confirmations, the visualization, the statistical significance of the confirmations, the binding energy, All that has to be taken into consideration to find out whether your virtual screening was a success or a failure. So you have this particular schematic here, it shows that you have a protein. This is a HIV reverse transcriptase. For many, many years, reverse transcriptase was thought to be the key enzyme in HIV for which drugs can be designed. And we have very good HIV reverse transcriptase inhibitors that do a pretty good job of eliminating the HIV virus from the body. But for a long time, they did not know that the active site is made up in a space that only when it is a dimer. So for a long time, they were looking at only the monomer. But when they looked at the dimer, that is when they found this particular cavity here, where these two residues, the active site residues, make an interaction. So this is the surface representation of the active site. So this is our area of interest. So you can find out the shape complementarity by many different methods. Here they have used different uh, spheres of made up of different radius, and you can fit a molecule that has a shape complementarity and makes an interaction with the active site and so on. So this is one scheme, or you can say step-by-step process. Here you have a receptor, you prepare the receptor, and then you fill it up the receptor to find out shape complementarity, and then you try to fit a molecule to see if that is closest to this shape complementarity or not. So this is a workflow of doc and how doc does a virtual screening or molecular doc. Before I go to Autodoc, if you have any questions about the basic of molecular docking, I'm happy to answer the questions. Yeah, go ahead. Don't have to wait. You can unmute and you can ask. Yeah.
Sir, suppose in GPCRs, they will the like that channels will be opened once they get a signal peptide. So can we for to my understanding, can I think that there the signal peptide can act as a ligand?
Can you repeat again? In GPCRs, the signal peptide goes and binds to it.
Like every for transportation of proteins, they need some signal peptide to transport them, and they will be located to particular targets. So suppose if I want to target any transmembranal or any like structural protein related to the plasma membrane, so for plasma membrane, what kind of ligands that we can design, like what might act as a ligand?
So if you take membrane proteins, you have two regions to consider. The first is the extracellular region, then you have intracellular region. The signal peptide that transports the protein to the exterior of the cell, it usually is coming to the intracellular part of the membrane protein. Intracellular part of the membrane protein is not accessible for an inhibitor that you're supplying from outside the cell. So in that particular case, your active site or the region of interest is the extracellular part of the protein. Okay. So once you know which is the extracellular part, then you have to see what is the area, the volume, and what type of hydrophobicity, hydrophilicity, charge composition, shape of those active site region. And that is after that, you can see, you can screen many different compounds and you can find out if something can fit into that particular place. Validation in that particular instance is if you have a residue that is critical for the protein's function in the outer membrane area, in the extracellular part. So does your molecule bind to that particular residue? Does it make a hydrogen bond? Does it make hydrophobic interactions? Then those criteria will tell you whether that is a good binder or not.
I really don't know. In cancer biology, to a particular cancer, there are so many there may be any signaling pathways or any structural proteins or any other metabolic pathways that can be targeted, but those pathways are also similar in normal cells, so we can only target those which are highly active and which are highly expressed.
Yeah,
Isn't that only, suppose if we target this, doesn't other normal cells also get affected, sir?
that is the whole point of an area of research called as subtractive genomics. So you have the genome of the human and you have the genome of the target organism. Let's take the common one, okay, like E. coli, which causes urinary tract infection. Uh, you have both genomes. Through evolution, we know that there are a lot of proteins in E. coli which share homology and structural similarity with proteins in humans. So if you're going to target the protein in E. coli, you might be targeting the host protein itself. So that is not good. So subtractive genomics, the area is to find out which are the unique targets present in the pathogenic organism, and any molecule that you design or you find out design a drug, it will be able to act only on that parasitic organism or the pathogen, not affect the host target at all. One example that comes to me is about 20 years ago, in the malarial parasite, they came up with, they identified a protein called the Duffy protein, which is present on the surface of the malarial parasite. And this Duffy protein doesn't share any homology with any of the known human proteins at that point of time. So many area of research, many years of research happened where they tried to target the Duffy protein and find out the molecule that can bind to Duffy protein and prevent the Duffy protein to take part in the life cycle of the malarial parasite. Okay, so again, coming back to what you described, the metabolic pathways, there are a lot of metabolic pathways that are shared between a cancer cell and a non-cancer cell. In fact, 99.99% of the pathways are same. And the entire area of systems biology, and in systems biology, you have the flux balance analysis, where you can find out how the flux travels through a particular pathway, whether you have alternate routes or not. So to give you an analogy, uh, tell me a city in India, wherever you are located, right? Let's say Delhi. Okay. Now, if you are going to travel from Delhi to Chennai, you can travel by air, by train, by road, or you can even walk. You can probably take the longest route. You can swim through the Ganges, come through the Bay of Bengal, and come to Chennai. There are many, many different routes. So, if let us say there is some issue and the major transportation is blocked, then do you have alternate pathways through which the flux can still travel through and still do its job? And those type of pathway analysis is a crucial step in target identification. So in that, all these is why it's called rational, because you have a logic. You are not just looking at the molecular mechanism, you're looking at a lot many more and then seeing that it makes a lot of sense to have this as a target and not others as a target.
Sir, I want to develop a kit to identify whether a person gets cancer or not.
The question is well intentioned, but it is not a good question. Cancer is a stochastic process. It's not a deterministic process. In the stochastic process, the cancer is a mechanism where the cell became highly robust, cell optimized its function, and it found a way out to prevent cell death. In that particular case, the the cell is optimal and cell becomes pretty um, highly robust to any perturbations that come from the outside environment. It just so happens that while this is beneficial for the single cell or the particular cell, it is detrimental for the organism as such. So the fitness function for the cancer cell may be very high, but the fitness function for the organism is not very high. So rather than asking a question whether a person will get cancer or not, you can probably rephrase that question in asking what conditions and what parameters will lead to a person getting a cancer or a type of cancer depends on this A, B, C, D, E. Okay. Usage of tobacco, smoking, and then drinking, and so on. Okay. So these type of things are not new, but we don't have a solution. Insurance companies would love to have such a solution because they would like to implement that and see if the premium has to be charged higher for a person who is probably going to get cancer or not. There's an ethical issue also added to it. Uh, Ignoring that, what you are talking about, the kit is more something called, we call these days as digital twin. Look up digital twin, and you can see that digital twin is nothing but exact same copy of me on the in the cyberspace, and everything is replicated. What I am, my body, my physiology is in that digital component, and you can try and change some parameters and see what is the likelihood of that person getting a cancer or that particular drug being effective, and so on. You can even have digital populations. So there's a lot of research being going on. Digital twin is something that has been picked up in the last 10 years in drug design as well. Because you can't do experiments on one particular person. In personalized medicine, you need a digital twin to see if that molecule will be effective or not, and then try it out in the real life scenario.
Like in my hypothesis, by screening the genome, like genetic makeup of an individual, like if there are any mutations or any kind of changes mostly present in the oncogenes or tumor suppressor genes or any other essential genes which are necessary for the cell division. Suppose if we screen any kind of biomarkers related to that, so can we identify whether a person is at risk, or there might be a chance to prevent before, because nowadays persons everyone are going to their like behavioral changes are too much. Everyone is going to ego. uh, Suppose if something, there is a fear that something is going to happen, so they will leave everything, they'll leave all jealous ego everything, and they will be very down to it. So that is one kind of
You
emotion.
are you're confusing sociological factors with physiological factors.
True,
Okay.
true, sir. Like
So
emotionally.
we are talking about physiological factors. We are talking about things that are reductive in nature. We are looking at the molecular level of an anti-cancer drug that can bind to a cancer target. Okay. So the other things that you have described, those are very difficult because they are basically anecdotal. You cannot do a randomized clinical trial on that and find out whether that is a factor or not. It's absolutely impossible. Because you cannot prove that. It's like a dragon in a garage analogy. Like five people come and then only one person says, I can see the dragon, the other four can't see the dragon. Then you can't prove it. The burden of proof is on the person who claims that I see a dragon, not on other people who don't see a dragon. So those things are pretty complicated in nature. You cannot. There's a fallacy in that particular logic there. Anyway, any other questions related to molecular docking before I go to autodock and then show you haddock? Okay. Yeah, go ahead. Someone asked something. Maybe at some point of time. Okay. So autodock is a popular tool. You can. It was developed in 1990s in the Olson's group. They have three different search algorithms. This is for conformational search, finding out how the molecule structure is present in different orientations, different conformations. So uh, this is a very good topic by itself. The reason is that it was a biology-inspired phenomenon using which they came up with the computational method to uh, identify the conformation. And again, it's used back in biology. So the one that I'm talking about is genetic algorithm. So genetic algorithm is nothing but, I'll just show you. Okay. So let us say you have binary creatures. So binary creatures are those that have zeros and ones, and they have 6 genes. Each gene has an allele, 0 or 1. And then just like how in chromosomes you have recombination events that happen, crossover, mutation, and then a selection. You can have different types of combinations of the alleles being created. So you can have 2 genes, one from each parent, and you can have an offspring that carries one of the alleles of that particular parent. This is completely biological inspiration, how chromosomes work. So when they looked at it, they found out that this is a very great way to solve an optimization problem. So in optimization problem, you want to find out the optimal solution in the shortest possible time. And if you have to go an incremental step of looking at each conformation of the molecule and then finding out the structure, it's going to take a lot of time. So what are the genes, like from this example, what are the genes in a molecule? You have the X, Y, and Z coordinates. So you have a conformation with one XYZ coordinate. Yeah, another conformation of the same molecule in another XYZ coordinate. You take some part of this, another part of this, and then you do a crossover. You mutate those genes or those rotational bonds with some variation. You reject some of those mutations. You accept only some mutations. Okay, so when you have this kind of a setup, what is happening is, like I said, the conformation search space, this is like a hill and valley representation. You can jump between different conformations in the valley, and you can quickly come to the energetically stable conformation for that molecule. So you can have many different genes. Apart from XYZ, you can have torsion angles between the bonds of the ligand. All those things can be taken up as genes. So inspired by biology, computational people, not computational biology, but computational or in computer science, they found that genetic algorithm is a very good optimization solution. And it just so happens in Autodoc, they use that particular genetic algorithm to find different conformations of the molecule. So Autodoc, this is exactly what it is. This is a hill, this is a valley. So in you can keep jumping between different conformations, or you can actually go from one part of the valley to another part of the valley by doing a mutation in the conformation. And so you have phenotypes, which are the conformations, and you have the parent, which gives a particular phenotype to the child, and that is because you are switching one conformation with the other, and so on. So you can use the Lamarckian genetic algorithm to find the lowest fitness function, and then map them back to the respective genotypes, and then you can see that within a short period of time, it finds the good answer or the good binder pretty fast. So what they do in Autodoc, it involves two steps. The first one is AutoGrid. In AutoGrid, it uses a grid-based approach. So this grid is made up of number of points. So you can have 10 points by 10 points by 10 points, X, Y, Z, all three coordinates. Between the points, the spacing is about 3.75 angstroms. So 10 points means 37 angstroms. So this grid size will be 37 angstrom cube. And wherever this grid point, so each black dots here is a grid point. So wherever this grid point comes closer, or wherever the atom of the protein or the receptor comes on that particular grid point, what are you doing is you're creating something called as a grid map. So the first task in AutoGrid is, or in Autodoc, is generating this grid representation. And then this is the map that is required for the ligand to identify which can be the complementarity in terms of energy, in terms of shape, in terms of conformation or binding pose, and then finding out how they how they can be placed with each other. So most commonly you have carbon, nitrogen, oxygen, sulfur, and hydrogen, for which grid maps are generated. You can have receptor-based grid maps, you can have ligand-based grid maps, you can have different types of grid maps. You can select one particular grid map for all the molecules and so on. So I'll go quickly to the genetic algorithm here. So this is the settings. These are the setups. You have 50 runs, meaning for each molecule, you do the docking 50 times. You repeat the docking, but the conformation is not the same. The information that you got is from the genetic algorithm, and you have initial population size. For each conformation, you can also have multiple populations. Here is the genetic algorithm parameters, mutation rate, crossover rate, and all those. And then what you do is that you find multiple solutions. Now you calculate the second part of the docking, which is scoring, and then you find out whether that scores are giving you binding energy. And the binding energy is the interaction energy in kilocalorie per mole. And you find out which is the most negative in number, because the lower the binding energy, the better. So more negative it is, the better. So from that binding energy, you can find out how many conformations among the 50 that you dog are similar. So you cluster them. So clustering is done based on either RMST. If you have a molecule as a standard, the control, then you can compare it with that. Or if you don't, then you can compare it with the binding energy within the 50 conformations itself. So based on that, you can have clusters of molecules. So remember I talked to you about blind docking, where if you don't know the active site, you give the entire protein and then you do the docking. So in that particular case, the grid is not this particular site. The grid is this entire protein itself. This entire protein is inside that grid box. And you see, let's say you do 50 dockings, you see how many of them will bind in this particular place. So let's say 30 out of 50. That means your statistical significance or the probability of the molecule binding in this particular orientation, in this particular position, is pretty high. Okay. So, and then if you have a comparison, you can see how close it is to the known molecule, and then you can calculate the RMST between that. Okay. So there are three different types of algorithms for the conformational search in Autodoc: simulated annealing, genetic algorithm, and local search. Simulated annealing is you are increasing the temperature to very high and then slowly letting it cool, which makes sure the steric clashes are removed and it makes a confirmation search possible. So that is something present in Autodoc. Most often we use Lamarckian genetic algorithm, but there's no hard and fast rule. Each of the methods are equally good. It depends on the particular system. System here is the ligand and the receptor that you're trying to find a molecule that binds to it. And then you're seeing whether it can bind in a particular orientation with a particular energy or not. So here you have the one in red is the original ligand. The one with the genetic algorithm or the local search is in green. The Lamarckian genetic algorithm is shown in gray. And then you have blue, and then the simulated annealing in yellow. Okay, so you can see that with each, you have different solutions. Which one is right or wrong? There is no such answer to it. It is only whether it is an optimal solution or a non-optimal solution. If it is not optimal, then you probably have to change the number of settings. If it's optimal, then you try to refine it and see whether you're able to recapture the same orientation by changing something else. So the logic behind this is that if a ligand is uh, expected or will bind to a receptor, it doesn't matter which method you use, you will find the orientation of that particular molecule bound in that particular receptor. That is for sure. So Autodoc in that way uh, runs on a different setup. There is another flavor of Autodoc, which is Autodoc Vina, where the this entire process is sped up, so it can be done at a much faster rate. And then there is a implementation of Autodoc Vina called Pyrex, Pyrx, and you can use that to find the conformations of the molecules um, in uh, in your system. Okay, so that is Autodoc. Uh, I am going to show you about. Yes. Wait, I had it. Let me just look at the browser that I had set up for Haddock. Here. Okay. Any questions? Please feel free to ask. Are you able to see my screen?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Okay. So Haddock is a tool that is present. It's basically an online tool. It's a very good tool for doing docking. And in Haddock, you do both protein docking and protein peptide docking and protein small molecule docking and so on. So the first thing that you need to do is you need to create a login password here, and it takes about 24 to 48 hours to get the password, the profile activated. Once it's active, then you can go to submit option. There are many different Haddock um, tools. Like for example, you can do cryo EM fitting, you can do data mining, you can do antibody based identification and so on. There are a lot many tools, but we are more interested in the Haddock 2.4. This is Haddock, the new version. It's not released. It's in the alpha testing. It will come after some time. So once you have it here, what you can do is you can go to submit a new job, and then there'll be a lot more interactive ways of doing this. Now, there are three tabs. You have input data, input parameters, and docking parameters, and so on. Okay. So you have to do step by step. So in the input data, you can give a name and then how many molecules you're trying to dock. So you can by default you dock two molecules, but then you can dock three molecules or four molecules. It will try to find maximum orientations between them. How many chains are there in the first molecule? Now, by default, the convention or the logical thing is that you are either doing a protein or protein ligand docking, but then hand dock, you can specify whether the first molecule is a ligand or a peptide or a glycan or a nucleic acid or some kind of a dummy object. Okay, so you have to upload your PDB file. Haddock doesn't take the PDB file from our CSV website and takes it here. Now, when you do molecular docking, let me show you the PyMol window. Are you able to see my PyMol screen? No, most probably not. Okay, let me share my full screen so that you see my PyMol as well. Okay. So in PyMol, I showed you about this, the B factor representation. Now, what you see when you load a protein molecule is that you will see the protein along with these small mosquito-like or fly-like things, small dots. Those are all water molecules because it's crystal water. These are not biological water, not the water molecules present in the cell. When we give the molecule or when we give the receptor for docking, you have to prepare the receptor file. So to prepare the receptor file, like for example, this is a sulfate, which came from the buffer when they did the crystallography. And you have something else like a glycerin or some kind of a glycerol or some kind of crystallization molecules, which are not biological in nature, meaning that they are not involved in the function of the protein. So the first task is to remove all of this and you get only the protein in uh, okay, so you only have the protein molecule, and here you can see that there is a disulfide bridge between these two cysteines. There are two disulfide bridges. This is fine until unless, let us say, this particular protein that I've shown you here is a copper binding protein. We know in this particular protein, there is experimental evidence, there is literature evidence that it binds to copper and copper is involved in the catalytic site where it does its function. Then I need to incorporate the copper in it, or I need to keep that metal ion in it. Sometimes magnesium, sodium will be there as part of the crystallization setup and you don't need them for the biological function. Then you need to remove them. And yesterday I was talking to you about stoichiometry. This particular protein works as a monomer. It doesn't require a dimer to function. So having one chain of this protein is enough for me to do the docking. This particular protein, I know that it doesn't take any water molecule as a nucleophile while doing the catalysis, and so I can remove all the water molecules from this. Similarly, phosphate, sulfate, everything. So you have to process your receptor file with only those that are needed. It's very rare that you would need a water molecule along with the receptor when you do the docking. The general rule of thumb is to just have the protein. Now, not all protein are complete, meaning some proteins, let us say this particular region, this loop region here, is highly mobile. And in the crystal structure or in the NMR, that particular region will be missing. And you can clearly see it in the PDB file where it will say missing residues. And if this particular missing residues is important for the function of the protein, then you have to fill it in the structure. You have to fill that gap and then use that structure for docking. All these steps are done to ensure that you are much closer to reality when you are doing the molecular docking. The more closer you are to reality, your experiments or the simulation is more valid and it is not imagination, or it's not or just a hypothesis. You will be able to see something. Predicted confirmation is the same as the crystal confirmation at some point of time. So you have to ensure that you know everything about that particular receptor before you prepare the receptor file for docking. So once you have that, then you can upload it to either Autodoc or HandDoc in this particular instance. And it will ask if your particular peptide or the second molecule that you're giving, which is a peptide, let us say it's a peptide. There are a lot of peptides which are cyclic in nature, and there are a lot of drugs which are peptide that are cyclic peptides and shown to be very effective. Amphotericin B is cyclic in nature. So in that particular case, you have to switch on this radio button here. If you want to do a conformational search, and if the molecule is too big, then you can coarse grain your molecule. Okay. Now, what is coarse grain? If you are able to declare, or you are giving every atom in your protein, then that is a fine grain up to the hydrogen. That's the smallest atom, right? So everything is declared, everything is given, then that is a fine grain. If you consider 20 atoms within a residue, within an amino acid as one single entity, then that is a coarse grain, because you are considering the hydrogens, nitrogens, C alpha, C beta, everything inside that particular residue as one entity. So that means it has very less flexibility for that particular amino acid. Then that is coarse grain. So you can switch on coarse grain if you know that that is some kind of approximation that you want. Okay. Most often in molecular docking, we look at atomic coordinates, we look at specific interaction at between two atoms at the hydrogen bond level, hydrophobic bonds, ionic bonds, and so on. So most often we don't use this particular step in HADDOC until unless you have a specific reason to do coarse graining. I would not recommend you to do coarse graining here. Then in the input parameters, you have to select in HADDOC active and passive residues. I'm not uploading anything here, but to show you, because it says that it is sequential, these setups are sequential, I'm going to show you what those active residues selection would look like. Okay. Okay, and let us take, let's go to PubChem. Everyone knows PubChem?
Yes, sir.
Oh, okay. So I'm just taking tetracycline as example. Okay, then I go to now. If this is a poor molecule example, let me. Okay, aspirin. That's great. Okay, go to aspirin, and then I go to the 3D. So it says you have to give it in PDB or MMCA format. So it's very easy to convert that. And I hope you all know about Babel. Anyone who doesn't know about Babel, Open Babel?
Yes, sir. It helped to
Okay.
convert the PDP to PDBT format.
Well, Babel is format conversion, any format to any format.
Yes, sir.
Okay.
Yes, sir.
So not just PDB to anything. It can be for anything else. Okay. So I give it as a PDB file. And so I converted the STF to PDB. Then there's a ligand. And then next, it will upload it. Multiple forms of the same residue. So this is a known issue. Now this is where you have to look at the occupancy. The serine 33. So there are two conformations, as you can see here. You can see a split, a small line here. So you have to process. This is part of the processing of the file, where you select only one conformation among the two known conformations. So this you can see it. So how do you see this in the PDB file? So you will see line 48. Chain B. So here you have these four lines in the PDB file where you have the one type of conformation for C beta, the beta carbon, and in the A and the B. So here you can see occupancy is 0.5, which means that there is equal likely chance of this beta carbon to be present in this XYZ coordinate and also in this XYZ coordinate. It's a good practice to have only one confirmation when you have this kind of occupancy. So to see if we have other occupancies, that you can see that there are not just the 33rd serine. You also have 131 serine in the B chain, which is also, and also the asparagine in the B chain that is having this type of a occupancy issue, where two conformations were seen and both were equally likely to be present in the average structure model. Okay. So in Haddock, what you do is, because it doesn't like this kind of thing. So it's again something Autodoc will ignore. Haddock doesn't ignore, Doc will ignore, Gold may not ignore. So it depends on the particular tool that you're using. And if it throws an error like this, that means you have to process your PDB file properly to do it. Nevertheless, the reason I uploaded that to show, or let us try with maybe the chain A. Let's see. Give me 2 minutes here. Okay, so there was a very poor structure that I took for an example, but let's go to the next part. I'll tell you what this is. So active and passive residues, what Haddock does is that your active residues, as the name suggests, are the residues where you would like the interaction to happen. Passive residues are the ones that you don't like it to interact to happen. So this is akin to saying this is the active site and these are the other parts which are not required for binding. So the active site region, what you can do to find in this particular interface, what if you upload a proper file, you will see a sequence representation of the protein, and you can select the residues. You can select a stretch of residues also. And on this side, you will see a three-dimensional image. You can rotate that and you can find out whether the selected regions are coming closer in space or not. So using this, you will be able to find out what the active residues need to be declared. And then in the second molecule, the active residue or the active in this, because I selected ligand, it will take the entire ligand as active, meaning any part of the ligand will bind to any the active part of the protein or the receptor. And then the third, once you have identified or declared it, then there are a lot of parameters that are there. What is the sampling parameter? What is the surface contact restraint? Then what is the clustering parameter? It also does the clustering based on common contacts, or you want to do it based on RMST. What is, if you're doing it RMST, what is the cutoff for RMST? You can play around with a lot more parameters here. To get an idea about what it does, you can actually go ahead with the default settings. Haddock is designed in a way, and this is true for even Autodock and for any other docking tool, that the basic default settings are quite good enough to do to get an initial idea of whether your docking is successful or not. But that is a problem with using default settings. When you use default settings, you consider the system as a black box, meaning that those general parameters that worked may not be the optimal parameters for your protein and ligand or the receptor and ligand. In that case, you have to play around with the parameters, you have to play around with the settings, and you have to find. Then you will ask the question, how do you know which is good, which is bad? Well, here's where the positive control will come very helpful. You take the positive control, you take the ligand or the substrate that is known to bind to the protein, you try to dock it back again, meaning you have a crystal structure with the complex, the protein and the ligand. You take the ligand and then you try to dock it again. Did the default settings capture the same conformation and binding pose and binding energy for your molecule? If it did with the RMSD of less than 1 angstrom, then probably the default settings is good enough for you. If it didn't, then you have to change one parameter at a time and see whether it will recapture the binding pose as per the crystal structure. It's only that particular setting is suitable for your setup or your system. Uh, in general, you cannot have a software or a tool that works for all setups. It is a guideline. So it may not be optimal for what is optimal for protein ligand may not be optimal for protein DNA or for protein. But HADDOC is pretty easy and intuitive that the basic default settings works most of the time, 99% it works without any issues. The output of HADDOC is that you get a cluster, and in the cluster you get a z-score, you get the binding energy, and how many components or how many conformations were there in that particular cluster. So which cluster to take? You have to go for a cluster that has a maximum number of conformations and has a very low z-score. Okay, so in that particular case, that will be the cluster, and it's usually the clusters are ranked in terms of z-score. So the z-score for the cluster 1 is most probably your result or the one that you want to consider. And you can download the entire cluster or you can download only that particular model. It will give you 5 best models in each cluster, and you can take from that the top ranked model, or you can look at all the 5 models and you can select which one is biologically meaningful, meaning that it makes sense to interact with the residue and find out whether that is what it is uh, expected to do. Okay, so this entire talk was just an introduction. I did not go into a lot more specifics about orthodox or haddock. But what I would encourage all of you is that I will share the link, create a login here, go to service, and then you will here on the top, you will have a login, create a login, and then wait till you get it activated. And once you have it, you don't need an academic email ID to get activated. You can use your personal Gmail as well if you don't have an academic email, and you can see whether you're able to play around with it. And any tool, don't consider it sacrosanct. Uh, it's something made by humans, and there are limitations to each tool. For your setup, for your purpose, whether it works or not is something that you have to decide, and you have to see whether it works and you're able to understand it. So just like you get a toy and you try to see everything in that and then see how it works, how it moves, and then take piece by piece and then reassemble it and see if the toy works back. Same concept with any computational tool. You have to see at which particular setup or which particular setting the tool fails, because you need to know the limitation, because you cannot take it by default, and those limitations will help you to find out whether that is good for your setup or not. Okay, whether that limitation is okay. In some cases, those limitations may not be okay. So you have to play around with it. With that, I'll stop. If you have any questions, happy to answer them.